So today we have the pleasure of welcoming Dr. Liam Rooney. He's a fellow of the Royal Microscopical Society and the inaugural Scottish Microscopy of the Year. His lab now investigates infection dynamics, antimicrobial resistance, and host pathogen interactions. He's the University of Glasgow, and he did his PhD at the University of Strathclyde. So today he'll be telling us about 3D printed lenses, which is a new technique for rapid prototyping and low-cost microscopy for the life sciences. So thanks so much, Liam, for joining us, and please go ahead with your presentation.
Awesome, thank you. So by training, I'm a microbiologist, but you'll be glad to hear that I'm not speaking about microbiology today. Instead, I'm going to take you through some recent work that we've been doing on 3D printed lenses. So the entire initiative was really focused on creating open additive manufacturing methods for microscopy to try and lower the cost for high precision, high quality lenses that are often the limiting factor when developing microscopes in low resource settings. So my kind of journey begins by looking at the kind of lens manufacturing throughout the ages, and surprisingly, lens manufacturing hasn't really changed in the last, couple of centuries. So we've been using the same grinding and polishing and sanding methods that have been used since the Middle Ages, where we take bulk quartz or glass elements, and then we iteratively grind and polish them down into either magnifying lenses or in some cases these reading stones, which date from the Middle Ages here. And this is a very subtractive method. It requires very specialist and very expensive machinery to create these lenses. It's often time consuming, but most importantly for us, it's restricted by geometry. So quite often lenses take specific forms, either having a convex or concave surfaces, but beyond that, it's quite difficult to generate high quality geometries that are more bespoke, simply because the machining that's required to do that requires significant investment to develop new types of subtractive machinery. So this is where we turn to additive manufacturing rather than subtractive manufacturing, because it's accessible, it's low cost. Additive manufacturing and 3D printing has been used extensively by hobbyists to create little figurines and action figures. And so we reasoned that we could use this low cost, low barrier technology to generate lenses. So there's two main families of 3D printers. There's the stereolithography based approach, where you have a liquid resin that's photopolymerized into a solid substrate. Otherwise, there's the fused filament fabrication approach. This is where you take a filament of normally plastic, you heat it up to high temperature, and then extrude layer by layer your print. So we chose to use the stereolithography-based approach, and the reason for that is that it's higher resolution, and you can print with clear, transparent substrates. We start off by designing the lens itself. We then 3D print and validate its performance, and then we characterize and apply it to bioimaging. And that's the kind of process that I'm going to take you through today. And then I'll talk about some of the new things that we're working on currently. So for those of you who haven't used a 3D printer before, this is a rough schematic of how they work. So the specific type of printer we have uses an LED array using ultraviolet LEDs. They are then irradiated onto a surface, which is normally an LCD screen, that then masks every layer of the printed structure as it's retracted and pulled from a vat of photopolymerizing resin. And once we have this printed structure, we then take it to a series of processing steps in order to render it transparent and have optical surfaces. So the printing process itself leaves behind a kind of sticky residue on the surface. So we wash that off and then cure using more ultraviolet light the sample until we get a hard set lens. The surface of that lens is quite rough, as you'll see in a moment, but we then smooth the surface of that using a spin coating operation on both the planar and the convex side of the lens, or the planar and the concave side of the lens, depending on the prescription. And going through this process, we had to validate the performance of the printer at every step. So we turned to the kind of general public for this. In the general public, people who use 3D printers as part of their hobby have developed a whole suite of tools that are not particularly scientific, but they work very well for characterizing the performance of the 3D printer and calibrating the settings. So you can see here on the left in the top, you can see these tests here are called the cones of calibration, and it's 2 rows of teeth on one side and 1 row of teeth on the other side if it works perfectly. If you have any kind of difference from that, then it means that you either need to add more exposure or take less exposure during the printing process. So you can see here on the top where it's failed, it's got multiple teeth on one side where we don't want it. But on the bottom here, where I've reduced the exposure setting, you can see that we have a nicely calibrated print run. We can also measure the resolution of the printer using, on the right-hand side, this Amerilabs Cityscape. And you can see here, I'll turn on my laser pointer actually, and hopefully you can see it. Hopefully you can see my mouse. You can see that the windows and the kind of features of the city are a certain size, and we need to make sure that the calibration is correct for the printer to be able to resolve and print these structures. So once we've dialed in the printer, we then turn to the lenses, and surprisingly, it worked quite well. So you can see here on the left-hand side, we have an unprocessed lens that's came straight off the printer. It's got a really rough surface, which makes it quite opaque. But after we take it through the spin coating process, you're left with this optically transparent 3D printed lens. And you can see the transparency is significantly increased once we go through the post-processing steps. So now that we can 3D print these lenses, we needed to ask ourselves, are they actually any good, and can they perform as well as a glass lens of a similar prescription? So we first of all looked at the characterization of the surface of these lenses. We wanted to know how smooth they were, how uniform they are, and how thick that spin-coated layer is on top of the lens. Because if we're adding more material, we need to know how that might change the optical prescription or the optical performance of the lens. Then we need to understand how it performs optically. So how does it transmit light? Does it absorb light? And does it autofluoresce? Because that then dictates the imaging applications that we can use the lenses for. In terms of their performance, we tested how they focus a beam, how they behave like a lens, and then translated that to bioimaging applications. So here you're looking at some of the early work that we did to characterize the surface. We used a variety of contact and non-contact dependent methods to surface profile the lenses. Here you're looking at a stylus profilometer. So here at the top, you've got the X and at the bottom, the Y trace along the surface of one of these lenses. And you can see the kind of topography. So it looks fairly smooth, but this only gives you a kind of single dimensional overview of the lens surface, whereas we want to understand the smoothness around the entire curvature. So we then turn to some optical methods and some interferometric methods to try and understand the surface curvature and the uniformity of these lenses. So I used a method here called interference reflection microscopy, and what that allows you to do is a label-free interference technique that maps the surface of the lenses. So basically how it works is anywhere within the sample volume, when you image with a confocal microscope, if you have a refractive index boundary, you'll generate a reflection of the incident light. And depending on how far apart two surfaces are from each other, you'll generate either deconstructive or constructive interference. And then you can see an IRM image below here of this convex surface, and you can see these radially symmetrical topographical lines that map over the surface of the lens. So if you were to interpret this as a map, you would be able to calculate the height of these fringes at every point of the lens surface, and you would also be able to detect defects in the lens surface as well. So we built an analysis script that allowed us to take an image intensity profile along this line of fringes, and then you can calculate the interference phenomena and then calculate that into the height above the cover glass. And that effectively gives you the surface profile along that measurement line. And we can sweep that measurement line radially around the entire circumference. And so we were quite surprised after all of this that we could actually form an image with these lenses. So we used a graticule here to measure the field of view of a microscope that had a 3D printed lens incorporated into it. And you can see here, for example, this is a 2.3 millimeter field of view, and it has reasonable illumination uniformity over the field. And then we applied this to a plant specimen. Here you're looking at a cross section of a stem of a basswood tree, and what we're looking at in the region of interest here is a linear profile through one of the sections here, and you can resolve structures down to the order of 6 or 7 microns, which is quite impressive. That's about the size of a red blood cell, and it really kind of opened the doors for us in terms of what we could apply these lenses to. So after we demonstrated their manufacturing and their quality, we then started to think of other applications that we could use these lenses for outside of their manufacturing. And we turned to creating 3D printed multi-element objective lenses. So currently objective lenses typically contain multiple elements that are stacked into a configuration to give a particular magnification, field of view, and resolution. However, we wanted to take the cost away from manufacturing these by 3D printing not only the barrel of the objective lens, but also the individual lenses that are in there. And you can see one of these lenses here. We also wanted to make this applicable to both bright field and fluorescence imaging. So to do that, we had to test the autofluorescence profile of the resin itself, or the lenses themselves, because if they were hugely autofluorescent, that might limit how we apply them. So here you're looking at a comparison of a couple of key wavelengths and their autofluorescence profiles. So as you might expect, these plastic resins autofluoresce in the UV and the blue regions, but they emit very little in the green and the red, which is quite important for bioimaging because the fluorophores that are most commonly used are things like green fluorescent protein, GFP, or an RFP, a red fluorescent protein. So for biological fluorescence imaging, a red and a green gap here is actually very beneficial. And so we looked at some more botanical specimens here, and we compared the performance of a 3D printed lens versus a commercial objective lens. So this is a 50 times magnification lens. On the left here, you're looking at the guard cells and the stomata of a plant leaf. This is the gas exchange complex that allows the leaf to exchange carbon dioxide and oxygen. And you can see in the 3D printed lens that you can nicely resolve these cell structures, and there's some internal detail within them. We also looked at cyanobacteria, where you could look at the septation of the cells within these chains. And then we looked at epidermal cells in an onion leaf, an onion prep. And you can look at things like the nucleus structure and resolve it well with a 3D printed lens. It also works in fluorescence mode. You can see individual chloroplasts here using 3D printed objective lenses. And again, you can see the kind of septation of these cytoplasmic compartments within this cyanobacterial chain. So we've managed to make the lenses, we've managed to make the objective lenses, so why don't we make the first fully 3D printed microscope? I should say fully has a caveat here, we're not 3D printing the camera or the light source for obvious reasons. So this is work that we published fairly recently, and it was picked up by a bunch of international news outlets. And I have to say that the community or the public reaction to this has been quite encouraging. Normally, I think we see the public maybe a lot of mistrust in science, and there's some barriers towards the public perception of science. But when we published this, we saw people on the internet, jumping to our defense, and kind of advocating for us, which was a really nice part of the story as well. And we kind of stood on the shoulders of giants here a little bit. So we based some of the work on some existing work from the University of Glasgow and the University of Bath, looking at the Open Flexure project. So the Open Flexure is effectively a chassis of a microscope that's been 3D printed, and the team between the University of Glasgow and University of Bath have been working on this for maybe the last 10 or 15 years. And basically, it's a 3D printed microscope body, but the optics in the existing system are commercial, and that is a limitation due to kind of restrictions around the supply chain. So we reasoned that why can't we build on this platform that already exists and has been proven, but exchange so that they have not only a 3D printed microscope body, but also the optics within the microscope are 3D printed as well. So a schematic is shown here where we have a 3D printed objective lens, we have a 3D printed condenser lens, and the entire microscope for the body and the lenses costs about £7 to manufacture. If you want to add in an LED light source and a Raspberry Pi camera, it takes the total cost of the microscope to £45. The 3D printers that we used for this were £250 for the resin printer. For the process for the filament printer, it was about £150, and then the spin coating machine was about £1,200. However, I have to say that this is an optional step, that you can get away with kind of sanding and polishing these 3D printed lenses, but spin coating would give you much more control and quality when it comes to the final lens product. So with our fully 3D printed microscope, you can see some raw data here on the left-hand side. This is a resolution test target. And you can see here that we're able to see some structure, we're able to form an image, but there's significant contribution from chromatic aberration. However, this is easily solved using an open source ImageJ plugin that corrects this chromatic correction, and you can see that we can preserve structures down to about 5 microns in diameter, which is excellent for cell biology or clinical diagnostic applications. We can also get reasonable uniformity of illumination over the field of view here, which measures about 1.5 millimeters, and applying this to some histological imaging, you can see on the left here a smear of some red blood cells from a patient, and you can see on the right a section of human kidney that's been stained with hematoxylin and eosin. So we can resolve individual red blood cells. This is important for looking at morphological changes in red blood cells that might be indicative of sickle cell disease or malaria or some other blood-borne infections. On the right, in terms of the kidney architecture, you can look at the kind of gross histological arrangements. So you can see the nephrons here that filter the waste from the kidney. You can see the intralobular artery here with some blood in it in the center of the image, which is quite impressive from an entirely 3D printed system. Those are small lenses, and the final question that I want to show to you is kind of how large can we go? So these resin lenses that I just showed you before, these solid immersion lenses, were a couple of millimeters in diameter, but we wanted to create a custom element that was large and that could enhance the performance of common microscopy modalities. So we created a large integrated lens biprism for enhancing the contrast in stereo microscopy. And basically, if you're familiar with stereo microscopy, it's commonly used in material sciences and manufacturing. It's used a little bit in the life sciences, but it's really limited by the optical performance in transparent samples. And that's due to the way that on the left here that I show, the illumination isn't optimally designed. So you can see that you've got a divergent light source directed towards the specimen plane, but it's not particularly focused on the specimen plane, and it doesn't serve to make the most of the imaging system. So what we reasoned is that we could improve the uniformity of illumination over the field of view, and that we could enhance the contrast by correcting this illumination using a single element. And so we designed an element that was effectively a plano-convex lens coupled to a biprism element. This would be quite difficult to manufacture using traditional lens manufacturing methods because there's effectively 3 optical faces that need to be perfectly smooth and kind of occlusion free. And so we turned to 3D printing to manufacture this because of the flexibility that affords us. You can see the finalized product here. I've actually got it sitting on my desk, and you can see the geometries of the final printed product. And when you compare the performance in terms of the illumination uniformity over the field of view, you can see a standard stereo microscope illumination profile. You can see both axes of the microscope on the left and the right hand side that I've color coded in pink and blue. So in the top row, you've got the standard illumination where you have 2 very distinct spots of illumination, which is not optimal. On the middle row, you can see that I've added some diffusing elements in the light path, and they do somewhat of a good job in terms of spreading that illumination about, but you still have very much a left and a right bias in terms of the illumination. But on the bottom row, when we add in our single biprism element, you can see that we correct the uniformity to a much better degree. We're using more of the light more evenly over the entire specimen area, and this should inherently give us higher contrast as well. And so we turned to look for specimens that are transparent, but they have some refractive structure in them that we could resolve. And so what you're looking at here is the scale from the surface of a goldfish. And these are really beautiful structures that have concentric rings that are kind of like rings in a tree that tell the age of the fish or the scale. And you can see here a magnified version of a region of interest that's imaged under, on the left, a standard stereo microscope, and on the right with the simple addition of our biprism element. And you can see that on the right with this biprism, you can see the enhancement in contrast that you achieve, which is about 67%. And you can see that these growth annuli, these structures are much more evident with the simple addition of a single 3D printed lens element. So I'll just summarize by kind of covering that 3D printing offers exciting prospects for low-cost fabrications. We can achieve 3D printed lenses that perform the same as glass lenses. We've shown that we can create the first 3D printed objective lenses for both fluorescence and bright field imaging. We can create entire microscopes out of 3D printed lenses with the same, and we can improve resolution and contrast using either large lenses or very small lenses. The next things that I'm working on in the 3D printing open manufacturing space are looking at medical devices, so printing medical implants for infection biology, diagnostic fluidic chips, and I'm also looking at creating, some of you will be familiar with the endoscope that was developed by Mark Pickering and his team at the University College Dublin. So we're working with them on creating imaging systems for infection biology, which is quite exciting. And I'll finish off just by acknowledging the folks involved. So these are a list of all the people, both from the University of Strathclyde, but also from the University of Glasgow, where I currently am. And if anything comes up after this seminar, like please fire me an email or contact me on Blue Sky or LinkedIn. And yeah, I'm really thankful for you all tuning in and listening. Thanks.
Thanks to you, Liam. This is very illuminating. And also, I personally wasn't aware of the variety of the different applications you have highlighted. So that's super interesting.